This is going to be the first installment in a series of videos in which we here at Shenandoah University try to provide some information regarding SARS-CoV-2, the virus behind the disease COVID-19. So we're going to start out with the idea of how do vaccines work. We need to first understand the immune system.
Now, I should note quickly that this is going to be a very simplistic view of the immune system, and for those of you that are interested, we will be putting up a subsequent video in which we go into the ins and outs of the immune response in a much more detailed manner.
Start out by understanding that the immune system is broken down into an innate and an adaptive immune system. And what's important to note about the adaptive immune system is that it has a memory. So it is actually able to remember past infections. And that's a very important trait because that is what we as scientists are going to utilize in order to effectively vaccinate. So if we look a little bit closer at the adaptive immune system, we would see that there are more or less three main classes of cells. We have our T cells, our antigen presenting cells, and our B cells. Now, again, each will be important for fighting off SARS-CoV-2 or the disease COVID-19, but we want to focus in for this talk just on the B cells or the B lymphocytes. And what is important to note about these cells is that they produce what are known as antibodies. Now, antibodies are going to be a small molecule that can be released from these cells that will bind physically to things like viruses. And that's important because when an antibody is able to bind to another molecule like a virus, that is the signal for the rest of the immune system to attack it. So how is this going to work? Well, it begins with a B cell known as a naive B cell. But in the case of SARS-CoV-2, what we can see is that when it enters the body, there will be naive B cells that recognize it, right? So that's true of everyone. And when that naive B cell recognizes that virus, it will bind to it via a receptor, so a cell surface protein that will physically dock onto that virus. And that's important because when that interaction occurs, it tells our naive B cell to start dividing. And when that cell divides, it can divide to form what are known as either activated plasma B cells or memory B cells. Now, our activated B cells, what they will do is they will now start producing millions of antibodies. And those antibodies will travel through the blood where they will bind to that virus and that will signal the rest of the immune system to attack it. Right? This is how we clear that virus once it's in our body. The memory B cells, what they're going to do instead is they're going to physically travel to your lymph nodes, which you have spread throughout your body. And once they're in the lymph nodes, they will hang out, right? So what they're really waiting for is a second infection by the same virus, right? And this graph here is kind of a classic graph that shows two things. It shows, number one, the response of the body in terms of releasing antibodies, which we see in red. So the red line is indicating antibody release. The dashed gray line is representing the amount of virus in the body. So with any viral infection, we have an initial exposure shown here. That initial exposure is going to lead to a rapid increase in the amount of virus in the body. In response to that virus, as we talked about, those naive B cells will recognize, they'll divide, and we'll see our plasma or our activated B cells start releasing antibody. And so we see this nice big peak in red, this first peak, showing an increased amount of antibody, which will lead to the destruction of those viruses, and that gray dashed line goes back down. Well, over time, it's possible that you can experience a reintroduction of that virus, right? Maybe you are around somebody sick a second time, and that virus re-enters the body. And so we get a secondary exposure. But notice, at the time of that secondary exposure, we already have memory B cells in the body because those memory B cells live for decades. And because we already have those memory B cells, as soon as that virus enters, there are already cells ready. Those memory B cells will immediately start dividing, forming lots of those activated B cells, and we see this massive increase in the amount of antibodies produced during that secondary exposure. So vaccines are going to mimic what the body does when the body gets infected a second time. So with a vaccine exposure, what we are doing as scientists is we are introducing virus, and we're introducing that virus either in a dead form or just part of a kind of broken apart virus. That's a topic that we will cover at a later time. But we introduce that virus, and what that does is it induces a large increase in antibodies, right, which we've already covered once before. But the big thing that the vaccine is doing is it's creating memory B cells. If you get exposed to the actual virus out in the actual world, right, so you get introduced to the actual SARS-CoV-2, we already have memory B cells that are present. So even though that virus is going to start creating more copies of itself in the body, you already have this large amount of memory cells, which allows you to produce a lot of antibodies very quickly, right? What this will do is it will produce lots of antibodies and quickly clear the virus, right? And that's what a vaccine does. So that way, when you get introduced to the virus in the environment, you have defense mechanisms that will help you quickly clear the virus. And the quicker and easier you're able to clear that virus, the less likely you are to have the symptoms of being very sick. So hopefully that video gave you a little bit better understanding of how vaccinations work, or at least the theory behind vaccines and why they are effective.